Hello and welcome to the Computer Vision Festival, hosted by the AI Accelerator Institute, a global community of over 400,000 AI leaders and practitioners, all striving towards one shared goal: inspiring the next generation of machine intelligence. With expert presentations on the festival agenda covering everything from space exploration on Mars to self-supervised learning, you're in for a treat. If you have any questions for our speakers or want to connect with them further, then ask away in the comment sections of the video and hear them answered at the end of each session in our live Q&As. If throughout the day you're loving the content, then be sure to check out the AI membership plans, where you can get access to exclusive content in the form of interviews, reports and articles. Access all areas passes to all past and future events. You can also join our Slack community, which is home to more than 2,500 fellow AI, machine learning and computer vision professionals, and is full of dynamic conversations and contributions. So if you're not already a member, come and join us today. Sign up using the link on the AI Accelerator Institute website. I want to say a massive thank you to all of our sponsors and partners for helping us to bring the festival to your screens today. Our amazing speakers for contributing some awesome content, and to you, our attendees, for joining us today. That's it from us, so go ahead, sit back, relax, and enjoy the festival. Okay, then we shall just wait a couple of seconds for everyone to enter the room. Just give it 15 seconds or so. And then we'll kick this next session off. But really hope that everyone is enjoying the festival. I know we've got quite a heavy 3 days, so please take your time to digest all the content. Okay, Gunther, it looks like everyone's in the session, so welcome back, everyone. Super excited about this session. It actually got one of the most registrations out of all of the entire festival, so pressure's on, Gunther. I'm sure you'll be fine. And let me just introduce Gunther Smits, the VP of Image and Data Scientist at AstraZeneca, and he is here today to speak to you about deep learning for digital tissue biomarker discovery in oncology. And on that note, Gunther, over to you.
Many thanks, Jordan, and many thanks for inviting me, and happy to look forward to the talk and the discussion afterwards. So just to give you a bit of context what we are doing in computational pathology here in Munich in Germany, and in translational medicine in general for AstraZeneca oncology. So as we've seen in the past years, maybe a decade, we've seen tremendous progress in oncology with respect to the survival rates of our patients with more modern drugs like IO, immuno-oncology treatments like antibody-drug conjugates, and there is a whole kind of new family of drugs which provide significant benefits for the patients. But on the other side, also we see that advancements in the therapy, still a significant portion of the patient does not respond to our drugs. And our task in translational medicine, and in particular in computational pathology, is to understand why those patients do not respond to the therapy and how to predict if they are going to respond or not. And that where deep learning comes into play, and in particular deep learning to understand images, histopathology images from patient samples enrolled in clinical trials, but further on also in the clinical routine. So I'm working for AstraZeneca, as it's disclosed in here, and as you know, and so therefore we may jump directly into the introduction, which is kind of very off topic, but which may show you some of our thoughts towards where we're aiming for. So that the machine may at some point in time surpass the human intelligence and show superhuman artificial intelligence. And maybe one of the most prominent events in the last year was in 2017, where DeepMind, the AlphaGo machine from DeepMind, played against the world champion in Go, KG, and really beat him to not death, but to just that he was completely shattered on ground. And And the interesting thing in 2017 is that the machine did not follow the route with IBM set up 20 years earlier with the chess software, which followed, well, just the computational power by looking into the future of the gameplay to defeat the human. But here the machine recognized patterns on the Go board, and according to the patterns it recognized, it played the game and played new moves the world has not seen before, which kind of introduced a kind of creativity invented by the machine, which was pretty new to this field. Therefore, I find this event very remarkable. And we can ask ourselves, well, it's a game, it's just a play. What's next? What are the specific applications of such kind of technologies? And a couple of years later, DeepMind again showed its excellence in machine learning. And with AlphaFold 2, they solved the long-standing 50 years standing problem to kind of predict the three-dimensional structures of proteins according to the sequence. So this machine learned that when you put in a sequence of amino acids into the machine, then it kind of replicates the folding process of these sequences to the 3D structure. And according to a, well, pretty much sophisticated system, which is not yet fully published, but there are some hints on how it works. It works with a mixture of unsupervised learning and supervised learning by using large databases with known structures and some physical modules. And from those, they could solve this problem and the challenge on how the folding actually works. Moreover, it speeds up the discovery process for novel drugs. So usually when people do that sequence or the structure prediction from sequences in lab experiments, they need a full year to do the experiment. And because a team of people doing that using crystallography and several computational methods to end up with this folding prediction. The machine nowadays can do it within a day, maybe. So it's a very powerful and machine which kind of accelerates the discovery of novel drugs by a factor of several hundred. So it's not just a factor of 10, it's not a factor of 100, it's much more. So that's really a big step forward for the drug discovery. But we are here to discuss the computer vision aspects, in particular for the deep learning applied on oncology images. But I have here a third highlight of the last year. So this highlight is back in 2012, where many people think the deep learning revolution started. And it's with Alex from University Toronto who invented and presented there in the ImageNet classification challenge. It's his AlexNet, what's nowadays it's called. It's a deep learning network comprising several tens of convolutional and pooling layers depicted in the middle of the chart. You feed in small images, image patches, and they are classified according to this convolutional network. The system has been trained with 1 million natural images with 1,000 classes. And at the end, for each image you feed in, you get a vector of class probabilities, where the most probable class really gives the final answer for the image classification. And what was remarkable in this achievement is that the classification error rate dropped by 10% compared to all other methods known before 2012. So it was a big step towards kind of understanding the images, not really, but kind of predicting the class labels of the images, and in particular with natural images and many, many classes, 1,000 classes. So in today's terms, the network is pretty small or reasonable. So it has 60 million parameters to adjust the weights of the neural network. Nowadays, let's say natural language processing and understanding models have billions of parameters. So they scale much larger. and could comprise much more knowledge than this ImageNet. But nevertheless, 2012, it was a remarkable success by Alex. So just as a refresher for everybody, maybe it's trivial for most of you, what's inside of such a kind of a network. It's the neuron. The neuron reserves input. This input may come from pixel values of an image in computer vision, but may come from other data like transcriptomic data, characters of a text document, whatever input signal you can envision. But most importantly, the input can come from the output of other neurons. And we have avoided some of all the inputs, do some kind of normalization at the end, and feed that output to other neurons. That's one option, or directly present the output as a result of the neural network. What people usually do, they stack all these neurons in layers, and with GoogleNet, for example, we've seen many layers down there, and therefore the name deep learning comes because it has a deep sequence of convolutions and max pooling and other operations, all using such kind of simple neurons. So that's the processing side. But one interesting thing for the training, which is quite frequently underestimated, is the optimization. So what's the aim of the whole architecture is that each input is processed in the right way. For image classification is that the network gives you back the right class label. For semantic segmentation, you get the right segmentation map back, and so on. So, and this optimization process in the learning is done by very advanced algorithms like the stochastic optimizers or Adams optimizers and methods. And so therefore, the crucial thing is what shall be optimized. So is it really a kind of an absolute thing so that we minimize the error rate for a given task? Or what was really remarkable with this Go playing architecture, that there was no absolute truth. So the machine played against itself, and from there it learned to become better, but it was relative. So it was just better than the competitor, but there is no, in chess playing, Go playing, there is no absolute. And this sets the playing thing a bit apart from the rest of the talk, where I'm talking about loss functions and optimizations towards finite goals. When we come to the biomarker discovery and diagnostics, I want to start from simple to a bit more complex. And the most simple thing and the most established thing we've seen with the AlexNet in 2012, it's a fully supervised approach. Supervised means that for each image or each object we see in the image, like a cell in histopathology, a human could provide a label. And when we look at the very small images, very detailed, which I show here to my usually computational pathology folks, we see individual cells in there. They are aligned in the tissue section, extracted from, so the biopsy is taken from the patient. The biopsy is kind of processed and cut and imaged. And you get large images, 100,000 by 100,000 pixels. And these small brownish and bluish images there, labeled as PD-L1 positive and PD-L1 negative, show you individual cells, which show some staining pattern according to the antibodies and the staining you apply to the tissue section. And there we see tumor cells and macrophages. These are immune cell populations and other immune cell populations like tumor-infiltrating lymphocytes. And all those cells play an important role in the prognosis of the patient if the patient is going to be treated with some immuno-oncology therapy, like with durvalumab or with other therapies, like nivolumab in this example. So from the task the system has to learn is to label the cells in the image in the correct way. And for that, Path AI, a company well known in the field, provided 250,000 class labels to a convolutional network that learned the kind of the knowledge represented by the pathologists and put into the system as class labels or little dots drawn into the image. And from that, the Path AI could derive a system which has been shown to act equivalent to the pathologist. So it does exactly the same if we do the correlation plot of the scoring or rating of the images done automatically with the convolutional neural network compared to what the pathologists are doing, it's equivalent. So this system provides a kind of a time saving, it provides more robustness, it works around the clock. Pathologists are a very scarce resource, so there is some issue with getting new people, young people into the field of pathology. So it helps the field of pathology, but it does not really perform anything new. It replicates things well known in the community since 10 years. But the replication is perfect. When we go to a next application example, that's one we are working on in Munich. It's around antibody-drug conjugate, and this figures has been, or the work has been published last year at the San Antonio Breast Cancer Conference already. So maybe first a few words to the biology, which is not that straightforward. So it's depicted on the left side in this figure. The so-called antibody-drug conjugate is a little protein, an antibody, which attaches to the surface of cancer cells, and it attaches to certain proteins which are present on the cancer cells. But it's like a Trojan horse. So when it attaches to the cell, it carries some warhead or toxic payload. It's depicted with these red dots there. And when the cell is usually very hungry and wants to eat as much as possible, so it internalized this antibody together with the warhead, and then the warhead gets released and kills the cell. So that's an interesting new kind of family of drugs. And what happens then when the cell dies? It releases also the warheads, which may go into neighboring cells and kill them as well. So to understand what's the challenge in here is that we have several processes in the play. So first we have to have these docking proteins expressed on the surface of the cancer cell. Then we have the ability, the hunger of the cell that to internalize, and then we have this neighboring effect with the warhead release and the so-called bystander effect that the neighboring cells are killed, independent of the expression of this protein on the surface. So nobody knows actually in industry on how to predict the success of a given ADC for a given patient. So when we have a patient in a clinical trial, we can actually not tell if he's going to respond to that drug or not. And that's the task we are working on. And for that, we trained a neural network, a semantic segmentation operating on two different levels. So first it detects tumor areas, and second it detects individual cells, cell membranes, and nuclei. And it does it trained by a pathologist. Again, it's fully supervised system, so the pathologists were asked to delineate individual cells and tell the system, and then we have a deep learning segmentation model to identify the cells. So that's all good, but from that we couldn't derive, it's not straightforward to derive a score, and therefore on this slide we tried various possible scoring methods, and as soon as you have the cell information at hand, you can play with bioinformatics nowadays very fast, different options for scoring the tissue slides and predicting the response to therapy of the patient. And the Kaplan-Meier curve on the right side, which shows you from a clinical trial, which shows you on the x-axis the progression-free survival in months, and on the y-axis the survival probability. There you see that the patients categorized in blue are have a higher improved response rate than those patients compared to the curve in red. And the most discriminative feature was actually one which considers the heterogeneity and not just of cell populations and not just the biomarker expression on individual cells. And just to remind, on each of those tissue sections, we see several 10,000, if not even 100,000 of cells. And the bioinformatics challenge is there at the end for the data mining. That was in the clinical trial. We have 100 patients with 10,000 cells in each patient, and to do all the data mining with spatial relationships very quickly and fast. A bit as a technical side note, and maybe you can go quickly over that complicated scheme, which was published, I think, last year or this year. Yeah, this year. And maybe you've all seen these figures of cycle GANs, generative adversarial networks, which kind of convert horses to zebras and vice versa. And to support our pathologists, we had the idea that we can transfer from one staining method to another staining method, and by doing that, save pathology annotation time. So it's a bit coupled to this question of supervised versus unsupervised learning. And here it's kind of a mixture that we use so-called chemical annotations. So the coloring of one stain on the left side, the yellow, the cytokeratin, which labels epithelial cells and tumor cells. We could transfer that one to the right domain, the PD-L1 staining, which is very hard to recognize and to discriminate tumor cells from macrophages and other immune cell populations. And that's then done automatically, because from the cytokeratin, the real cytokeratin images, we could derive or learn a system which generates so-called fake PD-L1 images, and the fake PD-L1 images are cycled back by the down loop to the cytokeratin domain. And if you do all right, then you transfer the annotations automatically from the cytokeratin to the PD-L1, and you save a lot of annotation time by the pathologist. So it's kind of a technical trick to use chemical annotations instead of human annotations, but it goes already into the direction of superhuman machine learning, because sometimes the chemical annotations are more reliable as the human eye in annotating. So, and the last sophistication we can think of in this field is the so-called end-to-end deep survival learning. We did this experiment in 2018 for gastric cancer, and there the task is to learn directly from the image content the survival of the patients without the need of segmenting the images into individual cells or tumor regions. So we just feed in individual image patches from multiple patients into the system, correlate those image patches with the survival of the patient using a loss function which is related to the Cox loss in survival analysis. And from there could derive a very nice and accurate stratification of the patients into long-living and short-living patients without the need of any pathologist support. So this machine is purely evidence-based, and as we've seen, it's in the previous talk with causality, it's not clear if the system learns a causal relationship between the images and the survival, or if it's just a correlation. So, and there is a huge debate what the system actually learned. And to resolve some of the learnings of the machine, we presented the so-called risk heat maps, so colorful overlays of the original images with indicating several risks, so a high risk of survival or low risk of survival, to the pathologist, and discussed the content and could derive from there some explanation what the machine discovered. But the challenge is here really the interpretability of the system by experts, and not the annotation of image content by expert. So the responsibility of the pathologist is completely shifted to the end of the analysis and the interpretation. So in this respect, we have to think forward on how to bring all this discovery work into the clinic and the clinical routine or in prospective clinical trials. And for that, we have to partner with the FDA or talk to the FDA, get from there the advice. And just recently, the FDA delivered a new guidance for software as a medical device. And when we read the document, it's pretty clear that one of the main issues they see with machine learning and artificial intelligence is the transparency of decisions, the robustness of the decisions, and the resilience to real-world artifacts and noise in the data and everything. And I think these are all things we have to include into the training, either by augmentation or getting other means to evaluate what the machine actually learned and get some estimates about the generalizability of our models. When we look at the industry landscape, which devices are already approved by the FDA and used in clinical routine, we see several in pink, which are related to oncology. Also, all of those not in histopathology, but mostly in radiology or non-invasive imaging. And all of those devices are kind of replicating what the pathologists or the radiologists in this field are doing. So they are not doing something new upfront from, as shown with the deep survival learning, but kind of easing the workload of the radiologist. And therefore, it's kind of straightforward to get the FDA approval. Maybe in histopathology, one of the most advanced systems in terms of regulatory approval is the cancer detection system for prostate cancer built by Page. And that's a pretty sophisticated system because of just the data they used. They used more than 40,000 whole slide images to develop and validate the system. And as said, each of these slide images, they are large images. They have 100,000 pixels in diameter. And therefore, you can train a lot. And most of the deep learning systems are using these image patches to be trained. And the image patches are pretty small, between 64 and 250 pixels. And therefore, in terms of image patches, the 44,000 whole slide images are tremendous data pool. So it's much, much larger than the ImageNet. And as you see, the challenge is not necessarily to train the classifier. The main challenge in the lower right row, it's kind of aggregate all the local probabilities of tumors to a diagnostic decision that the patient has the tumor and not an individual patch. And so therefore, for this, they use the two-step approach. So first classifying patches, and second, taking the 10 most risky patches in terms of tumor probability, and then classifying this bag to get the final decision for the patient. So they got this CE mark in Europe, but still they are waiting for an FDA approval. They have a breakthrough designation, but no real approval for the system. And maybe as a side comment, prostate cancer is the type of cancer which produces the most tissue sections in pathology, because usually when the patient gets it, is usually his diagnosis. Many biopsies are taken, so very frequently 16 and more biopsies are taken. So fine needle biopsies, 16 times punches in the prostate. And for each of those biopsies, they cut 3 sections. So we have millions of of images available for prostate, and therefore this makes it a good target for machine learning approaches, and again to decrease the workload of the pathologists. To summarize, well, as we all know, there are mainly three big drivers of the advancements we see in artificial intelligence and in particular deep learning. It's the availability of a lot of digital image data. Fortunately, some labeled image data, as we've seen in ImageNet, or with large pathologist networks like with PathAI. So big data is available, and modern methods utilizes few-shot learnings or other architectures may live with little data, and there are more advanced methods like AC GANs and unsupervised methods reducing the data load. But the initial big success of AI in the last years was really built on big data, parallel computing, cheaper consumer GPUs, and not to forget these novel algorithms, in particular the optimization algorithms to compute from the loss functions, from the optimization goals, the adjustments of the network layers for these individual neurons. And maybe one of the additional thing is just the swarm intelligence of the community. So we all kind of use the same frameworks to develop our software. It's at least we use the Google framework TensorFlow, and it's all open source, and therefore the availability of excellent software freely available, that's another driver for the success of AI. We are pretty sure, and we've seen with in radiology in particular, that AI will change the clinical routine. And we are still looking forward that the computer will outperform humans. It did not happen yet in clinical applications, but it's kind of on the horizon. And from our discussions with the FDA, they are very open to novel approaches using AI and machine learning in the clinical routine if we can robustly show that there is a benefit for the patient. With that, here are my acknowledgements. So first, the teams in Munich, so my research team from the AI research team and the data sciences and the pathology, helping us to annotate the data, but more importantly, to interpret the data. And then, of course, we are a global organization working across the ocean with AstraZeneca in Gaithersburg, and here the little ocean with Cambridge. And obviously, we also have academic collaborations around the world. Many thanks, and happy to take your questions.
Hey there, Gunther. So yeah, once again, massive thank you. It was a great presentation, as everyone expected. And we do have a couple of questions here, so we will turn it over to Q&A. So if you do think of a question, be sure to post it in the chat, and we'll get around to it now. First question comes in from Jazil. So just to clarify, so the performance is evaluated by the clinicians or radiologists because it is unsupervised. And did you face the lack of medical data or class imbalance problem while training? If so, how did you overcome them?
These are many questions. So I think you refer to the slide with the end-to-end learning. And there are many issues with the end-to-end learning. So most notably, you learn the class labels and not individual cells, which kind of restricts your learning data set to the number of patients in the trial. And to be specific, in this experiment, we used 200 patients. And to overcome a bit the limitation of the restricted number of patients, we used several augmentation methods. And of course, we looked into the class balancing, but it turned out in this experiment and other experiments that it's better to take the class frequencies as they are and not rebalance. So that was just our experience. We don't know exactly why. And I think the literature goes in all the directions, but usually we take the natural frequency of classical pulses to learn the system.
Okay, thanks, Gunther. So next question comes in from Josip. So as I can see through this presentation, most medical image classification methods are based on splitting the image into tiles. What is the reasoning behind that decision?
So the main reason is really the restriction of memory in the GPU. GPU memory is very limited, and the tiles are chosen kind of to fit into the memory of the GPU. The bigger the memory is, the bigger the tiles is. That's one question. One thing. The other thing is, the larger the tiles, the more weights you have in your network architecture. The more parameters you have to train, and the more complex the training is, the more training data you may need. So in little patches, you have a lot of samples to train your relatively small network. With large patches, you have lesser samples, but more parameters to optimize. So therefore, people tend to have smaller patches somehow, in particular in radiology, where we use three-dimensional convolutions. So it's three-dimensional patches, and those are becoming really small. So 30 by 30 by 30 voxels is rather the standard, or 60 by 60 by 60. And whereas here in histopathology, we have 200 by 200 up to maybe 1000 by 1000 pixels. But it's restricted by memory mainly.
Nice, really nice. Next question comes in from Naz Taran. So great work. How did you create your training data set? Did you synthesize them or obtain them from real data available in hospitals?
Absolutely. So in our daily work, we usually use biobank images to train the system and blindly apply the locked algorithm on clinical trial data. Sometimes we use kind of old clinical trials for the training as well, but usually we try to avoid to train on clinical trial data, but use biobank materials.
Cool. We've got another question from Jazil. So what is your opinion of developing something like ImagineNet for medical devices?
Pardon?
I'm just going to post it to you again so you can see it. So what is your opinion of developing something like ImagineNet for medical devices?
ImageNet. I think we are doing that in this example, or in all the other examples. Maybe the question is more towards having a public database available. And we have one available in the US. It's the TCGA, which comprises mostly H&E images, 1,000 and 10,000 of H&E images, which come with survival information and other diagnostic information, which is frequently used in many publications and academic work. TCGA from the National Institute of Health.
Nice one. Thank you, Gunther. So Wendy, thanks for posting the question again. So the question is, in terms of microscopy and cellular images, do you run into many barriers with low contrast and difficulty with edge detection, or are stainings the key to identification? And then just to clarify the question, she's wondering if your work focused more on mechanisms to identify markers in cells, or was this based primarily on given stained images?
So in translational medicine, usually we get for applications our images from clinical trials, and they are stained with contract research organizations using their standard protocols. And for that, the quality of the images is certainly far below what you can expect from a dedicated experiment in a preclinical setting. That's true, but nevertheless, we should think about where should a biomarker we discovered should go to. And finally, it's the clinical routine. That's our aim, to bring the biomarker to the patient finally. And therefore, it's for our purposes, it must be a robust biomarker working on a robust assay. and without too much sophistication, which works just everywhere in the world for every patient. And that's the main goal. Of course, we are doing some experiments in the preclinical world where we are utilizing multiplex immunofluorescence, for example, which sometimes provide much higher quality than the immunohistochemistry.
Cool. So next question is 2 seconds. So class imbalance is a major issue. Do you make augmentations to minimize the effect to minimize the effect?
Yeah, I said usually the class balance, we don't handle it since it has turned out we did all the experiments, so we had covered the class imbalance in some settings by augmentations, but it did not provide the quality increase we expected. So usually we're doing the augmentation more to enrich the data sets and make it more robust with staining variations, with location variations, with some other distortions like blur, for example. So you can simulate blur and provide the system with blurred images. So there are many, many options to use augmentation methods, and I would really recommend to use those.
Nice, great answer, Gunther. So we've just got time for one more quick question, and it's probably a bit more of a generalized question as well, so it's a good one to finish on. So what are the business opportunities that you see, Gunther, in computer vision with medical applications?
There are many, many applications. I'm just speaking for oncology, where we want to identify the patients which respond to the drugs, and that's of a tremendous value. Either for the healthcare system, it's a tremendous value because you don't treat the patients which do not respond. For the patients, it's a huge benefit. It's not just a commercial benefit; it's a benefit not to be treated and get all the adverse events and the costs associated with versus treatment. So identifying the right patients and associating the patients with the right drug that has a tremendous impact on the healthcare. Just one. If you see this Kaplan-Meier curves, for example, in this image, on the right side, you see that just 50% we have in this cohort 50% survival of the patients. So 50% of the patients do not survive within the study time. And all what we can do is kind to identify the right therapy for the patient has tremendous impact.
Amazing. Yeah, absolutely amazing work. Such a fascinating presentation, Gunther. So yeah, on that note, we'll wrap it up. Once again, a huge thank you to yourself.
Thank you for the invitation. Yeah.
No problem at all. You're welcome back anytime, Gunther. I'm sure you're going to be hearing from me again sometime in the future. But yeah, it's been a pleasure speaking with you, and a pleasure hearing your presentation. Thank you once again to the audience. And what I'm going to do is post a link to the next session in the chat now, as we have Intel coming up next. Thank you, Gunther.
Many thanks. Goodbye. Many thanks for the discussion.
Take care. Bye-bye.